This is module 1.2. We're going to have a bit of a general introduction to the things CompuCell can do, and then we are going to build a simple simulation together. And as always, this meeting is live streamed and recorded, so please be aware of that. We're going to come back and repeat a little bit of what we said earlier about why we need mechanistic models. I'm going to show you some of the recent work that's been done using CompuCell. And then we're going to run through very, very quickly some of the publications and ideas that CompuCell has been used for over the years, just to give you a sense of the different kinds of problems that you can solve with CompuCell. Those could be cancer simulations, those could be developmental biology simulations, vascular simulations, and toxicological simulations. And then in the second half, we're going to use TwidIt and Wizard to build a very simple simulation. And then we're going to discuss how to use that Wizard to create simulation. Wizard will help us build simulation frameworks, but in general, we're still going to have to do some hand editing of those frameworks. Really, this is going to be a simulation which we can build with Wizard only, so we don't have to do any hand programming. And we're trying to build Wizard up into something that will allow you to do more GUI-based model definition. But that's going to be for next year. If anybody wants to help us do that, we'd love to have the help. And then at the very end, we're going to start talking about how models are specified. That's going to be a lightning overview. The rest of the week is going to be teaching you how to do it. So don't worry if you don't follow all of that. The goal is just so that it's not totally unfamiliar when we come back to it later. So again, we're in this world of biology, the world of development, homeostasis in developmental disease, and we're thinking about what cells do. These kinds of models were developed initially to explore the biophysics of cell movement. More than 30 years ago, this Glazier paper was written when Francois and I were both postdocs in Japan. We certainly never thought 30-some years ago that people would still be using these methodologies today. I have to say, if we'd thought people were going to be using these methodologies 30 years later, we would have been more careful when we wrote that original paper. Sometimes you could discover that things have more life than you anticipated. So the moral there is be careful, because you never know when something will actually prove useful. The kind of modeling that we're going to do here has not found its way into biomedicine as much as some other models. The pharmacokinetic models that we talked about are used by every drug company in the world today. If you want an FDA approval, you have to run a pharmacokinetic model of the drug distribution and elimination of the body. And there are actually commercial companies like Certara or Simulation Plus that make a living doing that. Quantitative systems pharmacology is another area which computation is used in a lot. That works mostly at the molecular level, doesn't consider feedback so much. And there are companies that do solid mechanics simulations, for example, for diagnosing embolisms and aneurysms like heart flow, which have been very successful. The kind of modeling we're doing now is still relatively new in a biomedical context, although teams like the one at Moffitt Cancer Center in Tampa, Florida, and their offspring, for example, at MD Anderson, are using models of this kind, not with this particular software, but with other software packages that are quite similar, to do personalized cancer therapies. So this kind of modeling is getting into the domain of cancer therapy, for example, design, personalization. So as I mentioned, heart flow does solid biomechanics. They don't model individual cells. Of course, there are non-spatial AI and mechanistic models that are used. Molecular dynamics for rational drug design. Nova Discovery, which is a French company out of Lyon, has very interesting clinical trial models as well. And so ultimately what we're doing here is something that we hope will integrate with those other methodologies in the future. As I mentioned earlier, we need these kinds of mechanistic multicell simulations because of the multiscales at which biology occurs, and particularly the fact that drugs in particular act at a molecular scale. Measurements happen at a macro scale, and the scale we care about for medical outcomes is in the middle. And we need computation to be able to interpolate between those scales. We could use virtual tissues to understand basic biology, predict positive and negative effects of chemicals, design therapies, and so on. Some examples of this, and I'm going to run through very quickly some examples using CompuCell, would be why do blood vessels, which normally are behind the retina, sometimes invade the retina as you get older, causing blindness. Could we optimize therapies for diabetic retinopathy? We did a blood vessel simulation earlier. Those blood vessel simulations are actually quite powerful in a medical context. Why do some people have lethal overgrowth of the kidney, called polycystic kidney disease? Why does acetaminophen sometimes cause lethal damage to the liver when at other times it's safe? Or how does a drug treatment affect the spread of viral infection in the lungs? CompuCell is one of the more widely used packages. As I mentioned, there are quite a few of them these days. In 2024, these are papers that have been published so far. A few of the people who've written these papers are going to either here in the room or are going to be giving talks on their work. And we're looking forward to that. Every year we try to get some CompuCell users to come and present their models and teach people what they can do. These are 2023, but if you look at the titles of these papers, you'll see ovarian cancer, simulating cell shape, tissue invasion and epithelial monolayers. That's another cancer paper. Heterogeneity, so another cancer paper. Morphologies of microscale robotic agents, so more tissue engineering. Nephron progenitor cells, so developmental biology. Reaction diffusion theory of morphogenesis, more general theoretical understandings. Ductile carcinoma again, so more cancer as well. General simulation of how cells migrate, a model of drug treatment for SARS-CoV-2, design of molecular circuits for tissue engineering, simulation of dictyostelium discoideum development, unicellular to multicellular organization, so very much basic biology, breast cancer, zonation, liver zonation, so this is liver function and toxicology. And so you can see there are a whole variety of different applications that have been developed using CompuCell. So let's look at a couple of those very quickly. We're not going to have time here to go into detail, but I just want to give you a flavor for the different applications. You may come to this course knowing exactly what you want to simulate. That's fine. You may be just thinking about, could it possibly be useful to me? And in that case, perhaps seeing some different examples of the kinds of questions that can be asked could be helpful. A very old paper, which is one I actually regret we didn't pursue because it was quite successful at the time, was looking at the interaction between tumors and blood vessels. Tumors become hypoxic. They don't have enough oxygen in their core. They send out signals that cause blood vessels to proliferate. Those are the normal signals that maintain blood vessels and tissues. This is essentially a normal homeostatic or healing response. And in the case of tumors, it has adverse consequences, but it's a perfectly normal aspect of tissue biology and maintenance. And so we can ask what happens in that case, if you have blood vessel growth or not. In this case, we have a simple simulation. This goes back to 2009 by Abbas Sharinafard, who's now at St. Jude Children's Hospital, Memphis. We have a tumor. The tumor grows. We have oxygen and glucose supplied by the blood vessels. The core of the tumor becomes hypoxic. It sends out a signal that causes the blood vessels to proliferate. And you'll see in the right-hand case that proliferation is inhibited. The tumor still grows, it still migrates, but it grows more slowly and in a different pattern. And we can then compare the rate of growth. The early growth is the same. There's a period of stasis, and then there's a gradual divergence of the outcomes of the two simulations. The shapes of the tumors are different. The kinetics are different. The distribution of cell types within the tumors are different. One can do a lot of work of a simulation of that kind. There's a toy version of that simulation in the demos. We really need a better version. Another thing that happens is that you have what's called cancer progression. People think of mutations as causing cancer or cancer progression, but mutations could increase things or decrease things. Actually, mutations don't cause cancer. It's selection that causes cancer. And so you can ask the question, what's happening in the tumor microenvironment that's selecting for certain behaviors or phenotypes in particular contexts? And as tumor cells evolve, they change their environment, both with each other and with the extracellular environment. That means selection pressures change. And so you can run a simple simulation of a solid tumor and ask the question, for example, if the adhesion of the cells between each other and with the extracellular medium changes in time randomly, is there selection for certain kinds of patterns? And you can show that if there's a strong immune response, there isn't. The cells don't become less cohesive. They don't become more invasive. On the other hand, if you inhibit the immune response, you'll find that the cells, even though their adhesivity is changing randomly up and down, the ones that by accident have less adhesivity are favored in that environment. And you'll see that the net adhesivity of the cells will decrease and the net invasiveness of the cells will increase. And so you can actually then simulate with the avoidance of the immune system the result that you get metastasis. Now you could ask the question, how does the avoidance of the immune system evolve? That's not in this simple simulation, but it's something one could work with pretty easily. In developmental biology, we've seen this simulation before. This is our sort of prototypical simulation. It's the development of the segmental structure, the somites. In the embryo, there's a complicated set of gene regulatory and signaling networks running inside of each cell, network that determines the cell differentiation, signaling, changes of cell mechanics. But in this case, it was also shown by our experimental collaborators, Claudio Stern, that you could turn off the oscillator and turn off the signal for differentiation, and you still got segmentation. And so Giulio Belmonte, who's going to be lecturing later today, and later Priyam Adhyapok, worked to develop models of segmentation that were based on the mechanical properties of cells. And this is a simulation that one could do at the end of the week using more biomechanical detail of the cells. This is Priyam's simulation showing a traveling wave of differentiation that's going to lead to segmentation. In this case, we've used compartmental cells. The cells are divided into subregions and links to define their mechanical interactions. Giulio is also going to show you how to put molecular species within cells to allow molecular species to migrate within the cells. In this case, this is planar polarity in the evolving Drosophila wing disc. They're little fibers called hairs on the cells in the wing of the fruit fly that affect how the cell flies and also give signaling information. They need to be lined up. That lining up happens through a mechanism called planar polarity that involves migration of receptors from one surface of the cell to the other. And this is a detailed simulation of that polarization that's occurring in the developing wing disc. That could be coupled rather nicely to the calcium waves that our Notre Dame friends are working on. These are simulations of what are called our cell motility, using different kinds of representations of the cells. Pedro did these simulations at the top. Boyd's like simulation, self-generated gradient, which we'll do in the class. And then compartmental cells. This was a model that was developed by Gilberto, who's going to be lecturing for us. He can show people how to do this in 2 and 3D so that you can explore how cells move, how persistent movement in cells works, and all the very different phenomenology. Somebody was interested in bacteria, elongated bacteria interacting with each other. This third one here can be used to build representations of elongated bacteria and their interactions. We've worked a lot on retinal diseases and vascular diseases of one sort or another. Again, a beautiful paper, more than 10 years old now, by Abbas Shirinifard, looking at invasion of the retina by capillaries from behind the retina. This choroidal neovascularization after age-related macular degeneration is one of the main causes of adult blindness in places that don't have a lot of parasitic infection. In the third world, this isn't so common. In the US, it's quite a common disease. But when we started working on that, it was a bit mysterious because the blood vessels get into the retina, but in different patients, they go to different regions. And the prevailing theory that it was a growth factor dominated behavior didn't explain why you saw different invasion patterns. And so we spent a lot of time building a rather detailed simulation of the structure of the retina. Here's the Brooks membrane, the thick lining behind the retina. These purple cells represent the vasculature outside of the retina. Green represents the black lining of the retina, the pigmented retinal cells. Gray are the photoreceptors. Periodically, immune cells will punch holes in Brooks membrane. That's not unusual. Normally, the blood vessels don't invade. Occasionally, they do. Once they invade, the retina is always hypoxic, doesn't have enough oxygen, causes proliferation of the blood vessels. And in this case, the blood vessels proliferate, they cause retinal detachment and breakup of the retina and blindness. You can analyze what's going on, and as you age, the pigmented retinal cells adhere less to Brooks membrane. They don't adhere as well to the back of the retina, which is why you get retinal detachment more easily as you age. You can show that in the case of that impaired adhesion, you get precisely the proliferation of blood vessels between Brooks membrane and the pigmented retinal epithelium that's seen in elder patients. In some people, if you have trauma to the head, a car accident, you can get invasion of blood vessels between the photoreceptors and the pigmented retinal epithelial cells. That would be moving down this axis. If you get an infection of the eye, that reduces adhesion between the retinal pigmented cells and each other, and you get a different pattern of invasion. In each one of these cases, you could run these simulations many, many times for different parameters and calculate the probability of a particular kind of adverse outcome, a particular manifestation of this invasion. In this case, we're showing that if you have reduction in adhesion with the basement membrane, you have a high probability of this particular kind of invasion. And you can show that these other pathologies, well, these other stimulus, lead to particular other kinds of pathologies. This is called a risk map because it doesn't happen in every case. Each time you run the simulation, you get a different result. So it's a little bit like weather forecasting. You're really not doing prediction so much as forecasting. And that actually led to some understanding, new understanding of how this disease works and potential therapies. We've also used this kind of model to try to optimize diabetic retinopathy treatments. Starting in 2020, people became very interested in lung infections, viral lung infections. We actually worked with a bunch of undergrads and in a few weeks built a model of, very simplified model of a patch of lung tissue. You infect one cell with a virus, the virus spreads, the immune response comes in. Depending on the particular situation, either the immune response can prevent the virus from killing all the cells or not. This has a rather detailed model of viral replication in it. And you can use this then to analyze, for example, if you treated with an antiviral like remdesivir, or these days Paxlovid would be the choice. The outcomes are very different with different individuals, and depends a lot on how soon you start treating. One problem with antivirals is you really have to start treating right away, which is why it says after 3 days, there's no point in taking Paxlovid, or after 2 days, there's no point in taking Tamiflu. But even then, you get very different outcomes, and we were studying the variability of response under the same conditions here. Giuliano did a study, in this case, of the variability in response specifically for COVID antivirals, and was able to show that the spatial heterogeneity in the tissue meant you had to give higher doses and earlier doses than you would expect. You can study this also in vitro. You can infect a single small patch of cells in an in vitro culture that creates what's called a plaque. And you can study how that plaque develops as a function of various immune modulators. That's something that is a topic of still current interest today. You can build more elaborate models of the immune system. TJ took a very detailed model of the immune response to influenza, non-spatial model that had been very well calibrated in mice, and put it into a spatial context. Here we have many kinds of immune cells, many kinds of cytokines, and you can actually really look in quite a bit of detail at what happens in a patch of the lung during influenza infection. I should say that this kind of modeling, not using our software, but using their own software, has been used very extensively in modeling tuberculosis. So Denise Kirschner and her colleagues at University of Michigan have really advanced. This is a case, their multi-cell modeling work has really advanced therapies for tuberculosis, and it's worth looking at what they've done. Again, if somebody in a hackathon wanted to replicate some of their methods in CompuCell, that would be an interesting hackathon project. We've worked a lot in polycystic kidney disease. This is a little bit less familiar, perhaps, than cancer. It's also a proliferative disease. In this case, there are really no drug options. A kidney transplant's the only thing you can get. Your kidneys, instead of looking like that, look like that by the time you die. They keep growing. They form cysts. It's a pretty grim disease. People typically die in their 50s from it. It's caused, unlike cancer, which really is not a genetic disease, PKD is essentially caused by mutations in one of two genes, or both. If you have those mutations, you almost certainly are going to get the disease. If you don't have those mutations, you're not going to get the disease. The problem was we didn't really understand why a mutation in these ciliary proteins would lead to cysts. And Bob Bacalhau in our School of Medicine here in Indiana noticed that in these cystic kidneys, there was expression of a cell adhesion molecule, cadherin 8, that's normal in juveniles, but should be turned off in adults. And the question was, what was it doing? Now, cell adhesion molecules do a couple of things. They make cells stick to each other, which is important, but they also send signals that measure the degree to which the tissue is coherent. This is another homeostatic mechanism. When cells die in a tissue, which happens normally, or whether there's an injury in the tissue, you need to have cell proliferation to rebuild the lost tissue. If you have too much proliferation, you get a tumor. If you have too little proliferation, you get degeneration. And so what's called contact inhibition of proliferation is a core mechanism that gives local information about tissue density. The cell adhesion molecules don't just stick to each other. They send signals to the cell, telling the cell whether the local density is sufficient or not. And we could do simulations of different ways in which this cadherin 8, whose function was not really understood, could affect both adhesion and signaling about cell density. And here we built a simulation of a single kidney tubule. We've introduced a single cell that had cadherin 8 in it instead of the normal cadherins, because not every cell, even in the people who get sick, has the wrong cadherin. It's only about 1 in 1,000 or less cells that have the wrong cadherin. We were able to show that that single cell having the wrong cadherin was enough to form a cyst. And we were then able to do experiments in vitro where we put that cadherin into individual cells in vitro and show that they form cysts. All the cells in the cyst had the cadherin, the wrong cadherin. All the cells with the wrong cadherin were in cysts, and that matched this experiment and the simulation. We could show that other hypotheses about how it worked generated cysts, but with different morphologies. Question?
Yeah, so I had a question. So whenever the cyst grows in volume, are there any structural deformation or structural changes of the tube that it is growing on?
So in this particular case where we're simulating it in vitro, the answer is no. But in an adult kidney where there's a limited volume, yes, there are big changes structurally. But this simulation was really a very simplified one, trying to get at the mechanism by which the cyst formation happened.
In the case of the kidney, whenever there is cell proliferation, what kind of structural changes does it undergo?
Well, you'll see cyst formation, and you'll see disorganization of the tubule architecture because of it. The nephrons will be disorganized because there isn't enough volume. Things get crushed.
Is it kind of like an elastic response?
Yes, that's right. But if you actually look here, you can see the kidney has become quite disorganized. You see these large cysts throughout the kidney. If you had the pathology slides from these kidneys, you'd see that the kidney is really disorganized. Rita de Almeida, who's been helping as a TA this year, did some very nice, more classic bioinformatics analysis, trying to understand how this cadherin 8 actually mediated the cyst formation. And she was actually able to identify a set of drugs that potentially could prevent that. And those have been tried out in mice. It's a little bit hard to see here on the bottom right, but you can here actually see in kidneys in mice disorganization of the kidney's architecture. And treatment with these drugs then restores the correct architecture of the kidney. Kidneys in general don't have a lot of proliferation in them. They don't have much regenerative potential, which is one reason the kidney damage is essentially lifelong. It's a problem. Our colleagues are trying to find ways of getting that into clinical trials. Let's just say that. I mentioned computational toxicology. The EPA did a series of wonderful simulations over the years, some of which you'll hear about later in the course. Nicole Kleinstra, who is now head of NICEDM, one of the NIH institutes, when she was a postdoc, did some beautiful studies of chemical disruption blood vessel formation. That's a lovely paper going back more than 10 years now. Max Lung looked on genital formation. There was a lot of concern that estrogen in the environment could lead to feminization of the genitalia of animals and humans. And he was studying early development of the urogenital structures and showed how particular kinds of disruptions could lead to particular morphological failures in urogenital development. Shane Hudson, who was unfortunately away at another meeting because he's one of our best guest speakers. I strongly encourage you to listen to his talk from last year, which is on YouTube. Beautiful work on the formation of the roof of the mouth. The bones in the roof of the mouth have to fuse at the top of the mouth to form the palate. If they don't grow in properly and fuse properly, you have what's called cleft palate, which is a major and relatively frequent birth defect. And they were able to show in great detail both mechanically what's happening during fusion and failure, and also what the particular molecular perturbations and control of that are. So in each one of these, there's a detailed model of the molecular signaling and control of the cells, which allows you to model how particular chemical toxicants are going to lead to molecular perturbations, and then study at the tissue level how those perturbations are either compensated for or lead to failure of tissue architecture. A big area of research is liver toxicity. We worked on this ourselves some years ago. There's an enormous group using a different modeling environment, although very similar to CompuCell, but not CompuCell, led by Dirk Dresdow in Germany and France, now working in this area. And I strongly encourage people to look at Dirk's beautiful papers on this topic. In this case, we're going to build models of the kidney. I'm sorry, I say the kidney, I was talking about the kidney, the liver, excuse me. In this case, it's a very simplified model just of one vessel in the kidney. As I say it once, I'm going to say it wrong twice. In the liver, the sinusoids. In this case, we've got a model of the distribution of chemicals throughout the body, pharmacokinetic, pharmacodynamic model. And then we're going to take the box that says liver and open it up and replace it with a model of a liver sinusoid. And in this case, we're modeling acetaminophen for Europeans, paracetamol, which itself is mildly toxic, but which is metabolized in certain liver cells into a highly toxic molecule called NAPQI, NAPQI. It binds irreversibly to glutathione, which is one of the main antioxidants in cells. If glutathione levels get too low, the cell dies. And so what you're seeing here is actually an explicit model of blood flow. We don't do that very often, but you can do it in CompuCell. The blood is going to carry acetaminophen in it. That acetaminophen will be transported into the kidneys, to the liver cells, metabolized into this toxic byproduct, and then cause cell death. In this case, we're actually measuring the serum level, predicted serum level of the drug and its metabolites, and we're able to calibrate that against clinical data. And we can then explore what factors make some individuals more sensitive to poisoning by this drug than others. You can also build much more detailed models of the flow in the liver, which are pretty complicated, the flow, that is, and look at the microdosimetry question.
Yeah, so in the previous slide, you had that simulation of blood flow within a tube.
Yes.
So in that case, are you considering, so what is the nature of the flow? Like, is it laminar, or are you solving for the full navistones?
In capillaries, it's laminar flow. In the liver, the sinusoids have very small diameters, so it's capillary-like. The sinusoids are capillary-like, but they're rather strange. They're capillaries that are unique to the liver. They have a rather funny fenestrated architecture for the endothelial cells. The diameter is on the order of 10 microns to 15 microns. It shrinks a little bit towards the center, but not significantly. We actually did a whole series of experiments monitoring measuring those things. The liver work was picked up by a group at Michigan State University. that has modeled in detail what's called zonation. They've put in very detailed metabolic models of each cell in the liver as a function of its position. And that's very important if you want to know how long drugs last and want to know toxicity in the liver. And again, the European group led by Drezdo and his colleagues has taken that kind of work and really extended it a lot further. So I definitely encourage people who are interested in liver toxicity to look at that. Something that we've done a little bit more recently has been damage and recovery models. Joel, who's here on the call, and Michael Getz have been looking at eye damage. 2 billion people a day use shampoo on their hair. Some of them get shampoo in their eye, and that shampoo can cause eye damage. And you'd like to understand how that works. And the cornea has a rather interesting set of properties. It's a little bit like the skin, but the the stem cells that give rise to regeneration are all located at the periphery. And that means that the cornea has rather different damage and recovery mechanisms than other tissues. And we're going to build a model just now of the very top layer of the cornea. And we can here have a model of the periphery of the cornea. We're going to induce damage here. That's going to lead to growth factor ingression into the cornea. Interestingly, in the cornea, unlike almost any other tissue in the body, instead of having the blood carry everything, actually the tear film on the surface of your eye carries all of the signals. It's a little bit more like an insect where you have external flow than in other tissues. And you can do simulations of damage and recovery in this case. There's some beautiful work that's being done by Megan Haas, who I hope is going to be speaking to us on Friday, of muscle damage and recovery using CompuCell. She's particularly interested in the effects of estrogen on that. After menopause, estrogen levels go down in women, and they have much more difficulty with recovery after muscle damage. And so she's been doing really beautiful work trying to understand in detail how aging and estrogen levels affect recovery after injury in muscle. She's, I hope, going to talk to us about that later in the week. That's brand new. That paper just came out a few weeks ago in eLife. That is our overview, and then the next part of this is going to be a very simple exercise in using TWiT. Please fire up your CompuCell again. And I'm going to show you some very simple things to do in TWiT. And I'm going to walk you through it quickly, and we'll see how far we get. So the first thing is launch TWiT. You can do that by typing TWiT in your Finder and having TWiT launch directly. Alternatively, you can, if you have Player open on your screen, you can hit that pen and paper button on the menu bar in Player, or you can go to File and pull down Start TWiT. So I'd like everybody to get TWiT running. We're going to use Wizard, which is a simulation tool. So I'm going to walk you through it just this way. And then I can come back and do it in TWiT myself. But let me walk you through it first. See if we can follow along. So we have TWiT running. Everybody put TWiT on your screen and find the CC3D project pull-down menu at the top of the menu bar. Pull that down and select New CC3D Project. And you should get a dialog box that looks like this. It should say CompuCell 3D Wizard. Now, this is an old slide, so it has the word NanoHub in it. You're going to hit the Browse button and navigate to the place you want to put your simulation. Don't touch the simulation type box. We're going to leave everything as a default, pretty much. So But it's a good question. You're almost always going to select Python plus XML. TJ will show you what to do with the Python only one, but for the moment, we're going to leave defaults as they are, unless we need to change them. And again, we'll come back. There'll be lots of things we're not going to explain at this point because there are too many options to explain all of them now. But during the course of the week, we'll go through all of those options. So type the name of the simulation. A couple of pieces of advice. Absolutely don't start with a special character or a number, especially if you're in Mac or a Linux machine. Don't do that. No special characters in the name. No spaces in the name. Numbers are okay, but don't put them at the beginning. Okay. Secondly, in the browse, navigate to something where you can write. Don't point to a file that's not, to a directory that's not writable, and point to a directory where you want to have it. Again, it's a good idea to create for yourself during this course a directory, say on your homepage, called CompuCell Exercises or CompuCell Course. Okay, select the directory, click Next, and it will start creating a file which has a folder which has all of the model specifications in it. Some of the menus will look a little bit different on a Mac, and of course, what's readable and writable will be different in Windows, Macs, and Linux. And again, some names are allowable in Windows that are not allowable in Macs and Linux, which is why I would advise you don't ever put spaces or special characters in your names. No dots, no question marks. We had one person last year who really liked to begin their file names with numbers. Linux does not like file names that begin with numbers. Okay. Give it a name. You could call it My First Simulation or New Simulation, something like that. You're going to get a box next that says General Simulation Properties. We are going to skip that. Just accept the defaults. So hit Next. The next window will say Cell Type Specification. If we don't have cells, we're not going to have anything. So we need to create some cells. And so in the box, in the dialog box that says Cell Type, type the name of a cell. And again, don't begin the name with a number, no special characters, and no spaces. So cell 1 would be fine. And then hit Add. And then type cell 2 and hit Add. Could be normal cell and cancer cell would be fine. Any two names that are different, no spaces, no special characters. And if you don't hit Add, they're not added. So you have to remember to hit Add. When you have two, here's cell 1 and cell 2, hit Next.
What does the freeze function do?
Freeze will tell you to make the cells not move. So for example, if you want the cell to represent the wall of a dish, so it's non-motile, we would hit freeze. We'll use that a lot later, but not today. There are going to be a lot of options we're not going to explore today. This is just setting up the simplest possible simulation. Okay. Good question. Okay. And now you're going to get a window that says chemical fields. We're not using a chemical field, so just hit Next. The last window you're going to get will say Cell Properties and Behaviors, and this is the key one. You're going to select Contact, Growth, Mitosis, and Volume Local Flex. So make sure that you've checked those boxes. Contact, Growth, Mitosis, Volume Local Flex. There'll be some things here under Cell Property Trackers that will be checked. You don't have to do anything to those. You have to select these 4 boxes: Contact, Growth, Mitosis, and Volume Local Flex. If you fail to select one of those, the simulation will not crash, but it won't do what we want it to do.
In your slide, the last column, there are 2 ticks on the left side and only 1 tick on the right side. So I guess it's center of mass and cell neighbors. And cell neighbors is not ticked on the very last.
Right. Thank you very much, Katrin, for pointing that out. As I was just trying to say that the cell property trackers, depending on the exact version of CompuCell you're using, the defaults for what's checked and what's not are different. We're not going to be using those here. So if they're checked, it's fine. If they're not checked, it's fine. Doesn't do anything to the simulation at this point. In this case, the only reason that they look different is that the defaults got changed. And so one of these screenshots was done before the other. And so I didn't update the screenshots to be consistent, but you've got very good eyes for catching that. For sub-simulations, we will have to check those boxes. Volume local flex. Remember, nothing exists in these simulations unless we say that it exists. So if we do not say the cells have a volume, they do not have a volume. So volume local flex says the cell has a volume, and it's called local flex because we're going to be changing the volume of each cell locally, one at a time. Now hit next. And it'll say configuration complete, and you hit finish. And now you should see something that looks like this. It should say module. Though this is the one that I downloaded. This is the code that I have online in case it didn't work for you. You'll have the file name here in Twit, and you double click on that, and it will open up a little file hierarchy, and you'll see code. Try that. Does that work? We're not going to look at the code quite yet. We'll look at it in just a minute. Okay. Now, to save the simulation, you can hit this down arrow button here, or you can pull down CC3D project, save CC3D project. And there are a lot of save options, or you can hit this double old-fashioned floppy disk icon. I always recommend using that double saving everything. There are a bunch of save options. Some of those save options will only save certain components of the simulation, but the double floppy disk icon will save everything. So whatever you've changed will be saved. And there's actually a little bit of an indicator inside of Twit that will tell you whether things are saved or not. I find it hard to see though, so I don't tend to use it. Other people will explain that to you. Remember, when you use player, you can resize it and so on. Remember that in player, you can hit stop and start and step, as we did before. Do people remember how to run the simulation? To run the simulation, you can pull down CC3D project and select open in player. That'll work. Or you can hit the CompuCell player icon here. If you have player open from before, it may have the other simulation loaded. If that simulation is still running, it will ask you, do you want to stop and run the new simulation? Say yes. So this is a tiny bit of a test. Why don't people see, having generated the simulation, if they can figure out how to run it in player? I'm going to do this in Twit. First step, CC3D project, new project. New simulation 2. Next. Cell 1, add. Cell 2, add. Next. No chemical fields, next. Contact. Growth. Mitosis. Volume. Next. Finish. New simulation. Double click, new simulation, opens code, right click, open in player. Okay, simulation still running. Do you want to restop it and restart it? Yes. Hit step. Resize. There we go. And now I hit run. Okay. Let me go back to that. Here is Twit. In Twit, I select the name of the simulation here. I can go to CC3D project and say open in player here. Or I could right click on it and say open in player. And again, if I go to player and it's got something running already, it'll ask me if I want to replace it with the current simulation. Again, it's like riding a bicycle. The first times you do it, you think, how can I possibly ride a bicycle or driving a car? And you do it a few times, it becomes natural to you. And the problem is, once it's natural to you, it's hard to remember that it was hard. Most people saw something that looked like this. So you can try stopping it and restarting it. This is actually a very simple tumor simulation. It's an avascular tumor simulation. And in a few days, you're going to take that simulation, and in about an hour, you'll be able to build a pretty decent simulation of an avascular tumor spheroid, starting with this. The reason it's a tumor simulation is that the cells are not respecting any signal. They're growing and dividing without limit. In normal tissues, the signal between cells when they're pushing on each other prevents cell proliferation. It's called contact inhibition and proliferation. In this case,
oh, sorry.
Go ahead.
And now that we are talking on details, how the cells behave, wouldn't the mechanical forces bring the cells to die and to disappear? In this case, it's just disappearing, right? They don't are eaten up by macrophages or something like that. They just disappear when the pressure is too high, and then others reappear and so on, right?
That's right. This is not a very realistic simulation. The simulation early on, when the cells are dividing but haven't filled the domain, is not too bad. But in reality, we would have a pressure signal, a contact signal. There would be nutrients limiting the growth. We'd have a lot of other things that would regulate pressure. In this case, the cells simply grow at a constant rate and divide at a constant volume.
And this is just a box, so the cells cannot go out of this box. All cells have to be in this box.
Exactly.
Okay.
We can certainly add, for example, a real boundary or have the cells disappear when they get to the edge. All of these things are possible, but I wanted to have a simulation that we could build with just ticking boxes in the configuration.
Yeah, don't get me wrong. It's just to clarify what we are seeing, because usually when you have proliferation in a cell dish, you also see these circles. So cells are running in circles, and this is really interesting.
Yeah, absolutely. This is a very, very oversimplified simulation, and again, during the week, we'll talk about how to implement exactly the things that you've been talking about. Okay. Now, the last thing I want to do is just to give you a very quick sense of what the code looks like. And the rest of the week is going to be teaching you how to build simulations, which include contact inhibition, cell-based death of cells, and pressure-based death of cells, nutrient limitation, so on. Okay. CompuCell simulations, by and large, and again, there are always going to be some exceptions to this, consist of two main components. There's going to be something called CC3D ML, markup language, and Python. The static properties of the simulation are going to be specified in CC3D ML, and the dynamics are going to be specified in Python. And again, there's going to be a lot of this, but let's just very quickly show you how to see it and walk you through a few very highlights. Go back to Twit, and if you double click on the module or the name of your file, .xml, or the tab at the top that ends in XML, you'll pull up the XML part of the code. And you can use the scroll bar on the right. And for some reason, in current versions of CompuCell, that scroll bar is really narrow, so it's sometimes a little bit hard to grab. You can also use the up and down arrows. Okay. If you scroll through that code, you will see a top box that starts out with CompuCell 3D revision such and such. And then the first block will say metadata. And somebody asked about parallelization. The first thing it says is number processors. And that allows you to define how much parallelization you want to use, if any. It'll also have things that say dimensions, how long the simulation runs, a mysterious thing called temperature and neighbor order, which we'll come back to in the next lecture. Let's just look at one subcomponent to that. Dimensions says how big the box containing the cells is. And CompuCell by default doesn't have units. You have to convert it by hand. Typically, one voxel will be 1 micron or 2 microns or 5 microns. So you'll have to figure that one out. 256 by 256 by 1 is a 2D simulation. If I wrote 256 by 256 by 256, it would be a 3D simulation. Steps, again, time in CompuCell is measured just in steps. We happen to call them Monte Carlo steps, but just steps. And so typically one step will be a minute, but you can change that for your simulation. The next thing you'll see is the cell types, and we'll have a listing of the cell types. Medium is always there by default. It always has type 0. You'll have other cell types. Here we have cell 1 and cell 2. You'll see plugin. Plugins are a particular kind of component in CompuCell. If you're a computer person, you'll know what plugins are. They actually load modules into the CompuCell simulation. And those plugin, the cell types will show up as the cell type colors in the player window. The next component we might look at is something called contact. This is the contact energy, whether cells stick to each other or repel each other. And it's done by cell type. It's a little inconvenient. There's something called contact adhesion flex that's a little bit more biological. But here, the default is just that all the cells have the same contact energy. Okay, and that's here. And if you look in player, all of the things in the XML are going to be on the upper left-hand corner under Model Editor in player. And you can actually change any values in that as the simulation is running. So you can actually change the parameter values during the execution of the simulation if they're specified in XML, which is quite powerful. And we'll come back to that later. Okay. There's something called a steppable at the end of the XML. And we don't have a lot of initial condition generators, but you could draw a square box of cells. And you'll have steppable type uniform initializer that draws a square or rectangle of cells. You could specify the beginning and end coordinates of the rectangle, whether the cells are touching each other or separated, how big they are, and what kind of cells you want in the box. Okay. If we now switch to the CC3D Python by double-clicking on the name that begins steppables.py, or the tab steppables.py, you'll see Python code. Python code will have multiple subcomponents in it, which are functions. If you were on the classes yesterday, we had a class on classes. Python steppables are classes. You really don't need to worry about what a class is at the moment. There will be something called start function. That's called once at the beginning of the simulation. We use that to initialize things. And you'll see something like this: for cell in self.cell list. Cell is an object. Every cell is an object in the simulation. Cell list is a list of all of the cells. For cell in self.cell list, : does something to every cell in the simulation. And we're going to set a target volume for the cells and a lambda volume for the cells. The lambda volume is the inverse compressibility, and later today, Julia will start explaining to you what those mean. You'll see something called a step function, and it has an argument self comma MCS that you don't have to worry about at the moment. And in the step function, this is something that is done every time step. And you'll see again, for cell in self.cell list, doing it to every cell, self.target volume plus equals 1. Plus equals is a shorthand for adding to the value itself. And so this makes every cell in the simulation grow at a constant rate. It's not very biological. In reality, we would want that growth rate to depend on the nutrients, on the pressure, on the state of the cell, and so on. But this is a simple thing to do. The next thing that we're going to do is we're going to say the cells are going to divide sometime. We're going to create an empty list. If you were here yesterday, maybe you'll remember empty lists to hold the list of cells that are going to divide. We're going to run through all the cells. If their volume is bigger than 50, that's pretty arbitrary, we're going to say cell to divide dot append cell. So that's going to add the list of the cell to divide to the list. There's a Pythonic reason for this. In Python, when you're iterating over a list, you cannot change the identity of the list. If you have a cell divider disappear, the list of cells changes. And so it's important that you don't actually change the list of cells while you're checking what to do. Then we're going to go through the list of cells that we decide to divide. And now we're going to have a very powerful function in CompuCell, divide cell. And there are a lot of options. We can have the cell divide according to random orientation, fixed orientation, major axis, minor axis. If the cell's compartmental, we can have it divide, and so on. And the last thing we have to do when a cell divides is define what happens to the cell and its daughter cell. And there's a function update attributes. We say the parent cell has half of the volume that it had before, and otherwise the daughter cell is the same, clone parent to child. And then we have a little trick, which is if the parent was of type 1, we make the daughter of type 2 and vice versa. That's why you'll see the colors of the cells change when you run the simulation. That's it. Again, I realize that's a lightning view of things, but at least now you have a basic sense of how to create a simulation, how to look at what's in the simulation, run it, and a little bit of what Python and XML for CompuCell look like. And we'll be spending the rest of the week showing how those syntax of the XML and the Python work and how they relate to fundamental biological problems in CompuCell.
Yeah, so I have a couple of questions. So is it true that in CompuCell you can only deal with a Cartesian coordinate system? Like what can we deal with a polar coordinate and set the entire problem up in a polar coordinate or
No.
cylindrical?
So if you want to represent a circle, you'll have a lot of empty space in the corners.
And my next question is in the XML file regarding the addition energies. So I saw like cell 1 to cell 1 addition energy, medium to medium addition, and cell 2 to cell 2 addition.
Mm-hmm.
So I'm a little confused, like how is medium to medium addition energy? Like what does it mean?
So medium to medium doesn't do anything because medium is a single cell.
Yeah.
But if you draw a matrix, which is a n by n matrix, it makes sense to have the element there even if it's not used. That's a good thing to catch. Yes, medium doesn't mean anything. You could set any value there; it wouldn't make any difference.
Okay. Same as in like cell 1, and cell 2, cell 2.
No, cell one makes a difference because an epithelial cell will stick to another epithelial cell.
Oh, okay, okay, okay.
So it's by cell type.
Right. Okay.
Now
Got it.
now that only allows that means that all cells of a given type stick in the same way to all other cells of a given type.
Mhm.
The adhesion flex plugin allows you to have every cell have its own adhesion with every other cell.
Okay.
But it's a little bit more complicated to set up, so we're not going to show that until later in the week.
Is it true that when we specify the cell one medium addition energy to 10, let's say, so does that imply that I am actually specifying a kind of like uh, cell substrate interaction, like how
Right.
the cell interacts with the substrate?
That's right, yes. So if you think, I mean, medium can represent a lot of different things depending on the simulation. It
Uh-huh.
has fluid-like properties, so it could be literally liquid medium around the cells.
Uh-huh.
Or it could represent extracellular matrix, in which case the cell medium contact energy would be really representing integrin binding of the cells to the extracellular matrix. So
I
you have
see.
to decide what, in your simulation, you have to decide what that means. So I said you could change things that were defined in XML during the simulation. You can't go from 2D to 3D. Once you've defined that, you're stuck with that. There are options in CompuCell to change the lattice size. So you can there are calls in the Python that allow you to change the size of the lattice. So for example, if you had a cell that was migrating and you wanted to forget about the lattice over here and create new lattice over there, you can do that. We're probably not going to explain that during the class, but if you look in the manuals, you can do that. Switching from 2D to 3D, though, you can't do because the way energies are calculated and forces are calculated is different in 2D and 3D. So that won't work. You change that at the beginning of the simulation using that dimension tag, and that allows you to switch between 2D and 3D.
So I was wondering, like, is it true that since we went for a 2D framework, the cells were 2D, like 2D cells in a 2D framework? Suppose I have a 3D box and there are cells in there. How do I specify if the cells are 2D in nature or 3D?
So basically, that's determined by the lattice. If you set the lattice being x by y by 1, it's 2D. If you set it x by y by z, it's 3D. And the
Yeah.
cells will either be flat or will be round, depending on which one you pick. There's also, and this is new, and I haven't played with it much in CompuCell, the option of what we call 2.5D, where you have multiple 2D layers on top of each other, which can be very useful for modeling things like epithelia, infection in epithelia. And that
Oh, I see.
2.5D simulation. In the simulation that I showed of the virus infecting the cells,
Uh-huh. Uh-huh.
the layer of epithelial cells, which are really 3D, is represented just by their top surfaces, because that's where the infection happens. And that's a 2D sheet. And then there's another 2D sheet in which it represents the extracellular space where the virus is distributed and also the immune cells move. And it's
Okay.
much faster to do two 2D layers than to do a full 3D simulation. But since they're on top of each other, you can represent that as two 2D sheets that interact with each other. So it's
Okay.
possible to do that.
Okay. So I can choose any shape for the cells, like 3D, uh, 3D kind of like red blood cells and stuff like that. If my
That's
lattice is 3D.
right. It's just the 3D simulations are a lot slower because the simulation basically is proportional to the number of lattice sites. So if you
Oh,
write 256 cubed, it's 256 times slower than 256 by 256 by 1. So
yeah.
we typically, when we teach, we do mostly 2D simulations because they run much faster.
Oh, I see.
But if you're running simulations where you need 3D, for example, those kidney simulations, to make
Mhm.
cysts, 3D simulations are critical because the cysts really form in 3D. And so in that case, we ran full 3D simulations. But the reason we only had one tubule and a few cysts was because those are expensive simulations to run.
Yes. I'm very interested in the 2.5 like dimensions of the simulation, because in some cases, I know there are people, they are doing wildlife experiments, try to mimic the BBB, the blood-brain barrier. I think it can be helpful to help to simulate the BBB simulation, the model.
Tom Knudsen specifically done simulations of blood-brain barrier, though I don't know if he's going to present them this week. As I say, nobody comes to, I can't say nobody. If you're an expert in Physicell, then you know most of this stuff. Or if you've already used Morpheus a lot, you know most of this stuff. But in general, nobody comes to this class knowing everything. Some people have done more computing, some people have done more math, some people have done more biology, some people have done more medicine. And so, again, we go pretty fast, but the goal is not to assume that you understand everything. The goal is just to get familiar with the language and the ideas. And then as time goes on and you see things repeatedly, things may make more sense. Some things may be useful to you, some things may not. But don't worry if things are unfamiliar. If at the end of the week everything is unfamiliar, then we've failed in our teaching. But at the moment, it's like learning a language by immersion. You go into a classroom, people start speaking to you in German or Hindi, and at first you don't understand it. And then when you listen to it again and again, it begins to make sense. And we gave that list of possible readings so that as you identify, gee, I don't know something about developmental biology. I want to read a little bit about that. Oh, gee, I don't know Python classes. I read about that. Don't feel afraid if there's some things that you don't know. And again, people, especially in the hackathon, we try to team people up. So people with more computing work with people with more biology so that you can learn from each other.
So the model we simulated right now has an underlying governing equation. Where in the XML or the steppable files do I get the information of the underlying governing equations?
So you need to be a little bit careful. There is no single underlying governing equation here.
Okay.
There is a growth law, which specifies the rate of growth of the individual cells, which you could consider one governing equation. And there's another set of governing equations that describe the biomechanics, that describe how cells push on each other and interact when they're under pressure. And that's a second set of governing equations. And the image that you get is the result of the interaction of all of those different limitations. That's exactly what Giulio, we're not going to go into too much detail about that this year. Giulio will talk about that some. Some of the videos from previous years went into a lot more detail about that. And some of the online materials and some of the published papers go into a little bit more detail about that. Again, for some people, the physics is fascinating and they want to see all the physics details. For if you're an experimental wet lab biologist, the physics equations may be a bit alienating, just the way for the physicists, maybe long lists of gene names may be alienating.
Yeah.
So we're trying to balance out a little bit of biology, a little bit of physics, a little bit. And again, for everybody, don't be afraid if a few things are unfamiliar or you don't follow everything. The goal here is to learn how to speak a language. And you need some vocabulary, some grammar, some idea of structure, but you don't have to understand every word. And as you begin to speak, you'll begin to figure out what things you need to work on to learn more. So please, I understand it can be intimidating because there's so many things in the air at once. But I think if you're patient and we go through this together, by the end of the week, you'll have the basic vocabulary, the basic grammar, and you'll be able to write some simple models. And then as you've got one or two models working, you can then learn really quickly, because then as you modify things and extend them, you can explore. If you want to learn the physics, we can teach you how to do that. If you want to learn the biology, we can teach that. The goal here is to build a shared understanding. to begin with.